Welcome to today's Explainer. We're jumping right into the complex, fascinating world of epigenetics. We're going to untangle exactly how normal human physiology connects to pathological states and ultimately targeted therapies. If you're prepping for those grueling graduate pathophysiology exams, or hey, maybe you just want a really structured deep dive into how our genetic code is actually managed day to day, you're in exactly the right place.
Okay, here's our roadmap. We're treating this like a structured flowchart today. We'll start with the foundational cellular mechanisms, right? Then we'll move into genomic imprinting and environmental impacts, and finally, we'll scale all of that up to macroscopic diseases, cancer, and the pharmacological treatments we use to fight them. Let's get into it.
Section 1. Core epigenetic mechanisms. So before we can even touch the diseases, we have to define what epigenetics actually is when we're looking at our cellular machinery.
At its absolute core, epigenetics is all about changes in gene expression that do not involve altering the underlying DNA sequence itself. I've always liked the computer analogy here. Think of your actual DNA sequence as the hardware, right? The physical hard drive. Epigenetics is the software. It's the programming that tells the hardware exactly which genes to turn on and which to turn off, completely without touching that original source code.
And when we look closely, we see exactly how that software runs on a molecular level. Notice the three main mechanisms. First up, DNA methylation. This is basically slapping a methyl group directly onto a cytosine base, which generally silences or turns off that gene. Second, we've got histone post-translational modifications. These drive chromatin remodeling. See, your DNA is wrapped really tightly around histone proteins so it can actually fit inside the nucleus. When we add chemical tags to these histones, it changes how tightly that DNA is coiled. Super tight coiling, the gene is hidden and silenced. Loose coiling, it's open and ready to be read. And finally, RNA-based mechanisms are also in the mix, playing a huge role in regulating all this complex expression.
All right, shifting gears on our whiteboard, let's look at a really specialized area of normal physiology that relies heavily on these exact mechanisms: genomic imprinting.
Now, for most of our genes, we use both copies, the one from mom and the one from dad. But imprinting creates what we call monoallelic expression. That's where one parent's copy is permanently silenced through methylation. The evolutionary theory behind this, the genetic conflict hypothesis, is honestly wild. Basically, maternal genes tend to be imprinted to limit how many resources the fetus extracts, kind of saving mom's energy for future pregnancies. Paternal genes, on the other hand, are imprinted to maximize resource extraction and make the biggest offspring possible. It is literally a biological tug-of-war playing out at the epigenetic level. This same immense silencing power is also what drives massive cellular events like X chromosome inactivation in females.
Okay, pro tip time. If you're studying for advanced exams, you absolutely have to know this next contrast. It's the perfect example of what happens when imprinting goes wrong. We're talking about a very specific 4 million base pair deletion on chromosome 15. If a kid inherits this exact physical deletion from their father, they develop Prader-Willi syndrome, which brings short stature, hypotonia, and obesity. But if they inherit that identical deletion from their mother, they develop Angelman syndrome. Completely different presentation. We're talking severe intellectual disability, seizures, and an ataxic gait. Same physical deletion, totally different diseases based purely on which parent it came from.
You can really see how this plays out if you map it out. On the paternal side, usually the father provides the active genes for this region, while the maternal copy is naturally imprinted, meaning it's silenced from the start. So if that active paternal region gets deleted, the child has 0 functional copies left, and boom, Prader-Willi. Now, conversely, for a different set of genes in that exact same region, the maternal copy is the active one. Delete that active maternal gene, and the child is left with only the naturally silenced paternal copy. The result? Angelman syndrome.
The big takeaway here? The parent of origin dictates the disease. We see another classic showdown with chromosome 11 and the IGF-2 growth gene. In Beckwith-Wiedemann syndrome, an epigenetic loss of imprinting leads to an upregulation, essentially a double dose of IGF-2. This causes massive overgrowth, neonatal hypoglycemia, and definitely spikes cancer risk. Contrast that with Russell-Silver syndrome. Here we see the abnormal downregulation of IGF-2. That leads to profound growth retardation and a really characteristic triangular face. So again, same exact gene, opposite epigenetic errors, totally opposite clinical realities.
Moving down our flowchart, we need to look at how the outside world literally writes on our DNA. We're talking about environmental and lifestyle impacts. We've seen this play out in some pretty tragic historical events. Take the Dutch hunger winter of 1943.
Pregnant mothers were starving, and those severe in utero starvation conditions actually predisposed their children to obesity and diabetes decades later as adults. We see similar things with in utero ethanol exposure. The mechanism here is just mind-blowing. These early intense exposures can physically alter the activity of DNA methyltransferases, the very enzymes writing our epigenetic code. They lay down these abnormal marks that stick around and manifest as adult diseases. It's undeniable proof that our DNA sequence isn't our absolute destiny. Our environment is actively writing the software. It's exactly why identical twins who share the exact same hardware can develop completely different diseases as they age. Their unique environments program their epigenomes differently over time.
But sometimes, the problem really does start with the DNA sequence itself, which then triggers a whole avalanche of epigenetic errors. This is what we call a genetic and epigenetic dual hit.
Let's look at two perfect examples of this dual hit in action. First, Fragile X syndrome. This starts with the genetic mutational expansion, just way too many CGG repeats. The cell senses this abnormal expansion, panics a bit, and responds with dense, aggressive hypermethylation. This epigenetically silences the FMR1 gene, leading to the condition's neurological deficits. Now, flip that around for facioscapulohumeral muscular dystrophy, or FSHD. That starts with a genetic mutational contraction, a deletion. But unexpectedly, this deletion causes an abnormal loss of methylation, which inappropriately wakes up a gene that definitely should have stayed silenced.
And speaking of inappropriate silencing and activation, this brings us right to cancer. We now know that epigenetic dysregulation isn't just a side effect, it's a primary major driver of malignancy.
Tumor cells are incredibly, terrifyingly clever. They typically use a deadly 1-2 epigenetic punch. First, they trigger global hypomethylation. They essentially strip away those silencing methyl tags all across the genome. This improperly wakes up oncogenes that drive reckless, out-of-control cell division. At the exact same time, they use localized promoter hypermethylation. They actively add dense methyl tags specifically to silence our natural tumor suppressor genes. Clinically, we see this when the RB1 gene is epigenetically silenced in retinoblastoma, or when BRCA1 is silenced in certain familial breast cancers. And remember, this is all happening even without a traditional DNA mutation.
Okay, but here is the good news. Honestly, it's the most exciting part of this explainer. Unlike a genetic mutation, which is pretty much hard-coded into your DNA forever, epigenetic marks are just chemical tags. And chemical tags, we can potentially erase them. This opens up a massive door for targeted pharmacological therapies.
Take a really close look at the chemical structure of 5-azacitidine here. With a normal cytosine base, a methyl group is easily added by the enzyme DNMT1. But look at the drug version. By simply swapping out one specific carbon atom for a nitrogen atom, this drug turns into a trap. It irreversibly binds to DNMT1, totally taking that enzyme out of commission. So as the cancer cells divide, they experience a passive loss of methylation. The abnormal silencing marks simply fade away, and the patient's own tumor suppressor genes actually wake back up. We are actively using this right now in conditions like myelodysplastic syndrome. It's absolutely brilliant.
And we aren't just targeting DNA methylation, we're going after the histones too. Remember histone deacetylases, or HDACs? These are the enzymes that crank up chromatin compaction, winding the DNA so incredibly tight that tumor suppressor genes are completely hidden. So what do? We deploy HDAC inhibitors. These drugs block that extreme compaction. They force the DNA to stay loose and unwound, allowing those critical silenced genes to finally be transcribed again. This approach is showing some very real, very promising clinical results in treating breast and prostate cancers.
Which brings us to a pretty profound thought to wrap up our whiteboard session today. We've just seen how incredibly dynamic the epigenome is. We know it's shaped by our environment, and it drives everything from normal health to devastating disease. So if we can already use pharmacology to erase epigenetic marks and treat advanced cancers today, could we eventually map and safely reprogram the epigenome in utero? Could we literally stop these pathologies before they ever even begin? It is a massive, profound frontier in pathophysiology. Thanks so much for joining me today. Keep studying hard, and I'll see you in the next explainer.